we are going to have 2 lectures and discussions. The first is about biological sampling, and the second part is taxonomy. So each session has a lecture followed by Q&A session. And the first session will be led by Dr. Luciana Genio, and the taxonomy will be presented by Dr. Jinope. And tomorrow we're gonna have the gender mapping sections and biological information. And after that, I will assign presentation groups for action plan. So each of you will pick one of 2 topics, which is taxonomy identification and description. And the second group is the biological information. I will respect your preference. So at the end of tomorrow, just let me know what you prefer. But if when the group is really unbalanced, so it's not able to split, there I will assign some of the participants to the other group based on your expertise and work experience. And on Thursday, we're gonna have breakout session for action plan. And Friday, we'll have your presentation for action plan. So do we have to wait still, or are we good to go? Fiji office, is there anyone from Fiji office? Yeah, can we start, or do we have to wait on more?
There's still 3 hasn't came in yet.
Okay, so then we'll wait a bit.
Just about 5 minutes more.
Sure.
And if they don't arrive after 5 minutes, you can just go ahead.
Sure, okay. Then why don't you watch some short video about Koika or Mavic?
Did you know? 80% of all life on Earth is in the ocean. But we only know 1% of them. For greater knowledge leads to better conservation, and deeper research leads to sustainable uses of resources. National Marine Biodiversity Institute of Korea is seeking knowledge of the remaining 99%. There has been global efforts for conservation and sustainable use of the marine biodiversity. To join the cooperative effort, the National Marine Biodiversity Institute of Korea was founded in 2015. Through marine bioresources surveys, conservation, management, utilization, and promotion of public awareness, MABIC is striving toward conservation and sustainable use of marine bioresources in pursuit of common good or public good. MABIC is seeking value for Korea in the ocean. As a first step, the territorial waters are researched to locate marine habitats. Once a habitat is found, bioresources with potential as strategic resources are secured. And their information is shared with researchers and the general public via the Marine Bioresources Information System, or MBRIS. MABIC has collected approximately 7,000 species of marine life so far and is storing their specimen in its storage as a means to enhance the efficiency of its marine bioresources research. The MABIC explores ecological characteristics of vulnerable and endangered species and monitors changes in marine ecosystems regularly as a way to ascertain sustainability. Furthermore, MABIC makes efforts to discover alternatives to the resources imported by the bio-industry to domestically source the materials. MABIC also maintains close partnerships with relevant institutions through information exchange and joint surveys and research. Marine bioresources have high utility and are integral to the industry, and their potential value can be realized to the fullest. Genes involved in hemostasis and ovulation were discovered in the long-arm octopus, and genes concerned in tissue regeneration and respiration were found in Periophthalmus modestus. Marine genetic resources are being secured as such through marine bioresearch. MABIC systematically searches strategic resources with high potential for commercialization and performs genome analysis to discover candidate materials for healthcare, regenerative medicine, and other areas. A genetic modification detection kit has been developed to prevent the introduction of genetically modified Atlantic salmon and ornamental fish into Korea's territorial waters. MABIC is paving the foundation for the marine bioresources information to be utilized with efficiency. Relevant information that has been delegated to universities and research institutions has been integrated into the MBRIS to provide one-stop search services, and approximately 10,000 pieces of data are being provided for research and commercialization purposes. The Marine Biobank has been set up to distribute DNAs and tissues of marine organisms and genetic information to researchers in the industrial, academic, and research sectors to further promote the quality of marine bioresources research. Seacurium, a marine life museum, is being operated under MABIC with the aim of raising public awareness of marine life. Upon entering Seacurium, visitors are welcomed by the Tower of Life, which is the symbol of MABIC. The museum provides a chance for visitors to explore the world of marine life and learn about it easily and joyfully. There are diverse marine life specimens on display on the 4th floor, while a massive whale skeleton catches everyone's eye on the 3rd floor. You can explore the ocean in the Ocean Theater on the 2nd floor, and even at the Special Exhibition Hall and the Sea Snake Research Lab on the 1st floor. There are also a wide range of education programs for all age groups to boost their understanding of marine life. Furthermore, MABIC plans to establish a Marine Ecology Learning Center and offer even more outdoor experimental learning activities. Like the ocean that connects all 6 continents of the world, MABIC has formed a network with institutions worldwide under the common goal of further advancing the research on marine bioresources. MABIC, in compliance with the UN Convention on the Law of the Sea, Convention on Biological Diversity, Nagoya Protocol, and more, is collaborating over marine bioresources. And as the leading institution for researching overseas marine bioresources, MABIC has been forming partnerships globally, starting with the opening of a joint research center with Vietnam in 2017. MABIC will continue to engage in research and international cooperation to strengthen Korea's sovereignty over marine bioresources. 80% of all life on Earth is in the ocean. We only know 1% of them. To the other 99%, beyond the 1% we know, National Marine Biodiversity Institute of Korea will continue to research.
Okay, I think it's a good time to start. So like I said before, today we have 2 lectures, biological sampling followed by taxonomy. The first section is biological sampling. So here, I would like to introduce my wonderful colleagues and collaborator, Dr. Luciana Genio from International Seabed Authority. She is a rare expert in biological sampling, especially in deep sea area. And also she is playing a pioneering role in such a wonderful new project. It's called Sustainable Seabed Technology Initiative. So today, she's going to share her expertise in biological sampling. So please welcome Dr. Luciana Genio. And Luciana, mic is yours.
All right, thank you very much, Dr. Yang, for that very nice introduction. I'm sorry, it's a little bit dark where I am. I hope you can still see me well and hear me well. But I think the slides will show with the regular hour. So I will, yeah, while I put up the presentation, I can share a little bit. Wait, maybe I cannot do multiple things at the same time. So one thing at a time, maybe. Because I wasn't able to connect using the regular app. So I'm connecting through the, okay, I'm connecting through the browser. So now I'm having a little bit difficulty finding my presentation. Wait a second, maybe this is just, sorry about that. I hope this will, oh, maybe if I just open the presentation here, you will see it. Let me try. So what do you see at the moment? Oh, here we go. Okay, can you see the presentation?
Yes.
Okay, okay, perfect. Okay, so I cannot see you. So I'm just gonna trust that this is gonna all work well, and then we'll have some time to discuss after I present. Oops, why is this moving too fast? So yeah, first of all, I just wanted to say I'm very happy to be here, very pleased to participate again in this very interesting program that MABIC and KOICA have initiated, and as ISA has also contributed last year and this year. So as Dr. Yang said, I'm biologist by training, and I specialized in deep sea ecology. So I had a bit of experience with seagoing or several cruises offshore. And I'm going to share some of that. And in particular, in my current position as environmental analyst at the International Seabed Authority, I can refer to the importance of the collection or the sampling and the preservation, and in the context of the work that the ISA is doing. And even though this or some of what I'm going to say may sound quite distant from the reality in terms of more shallow or coastal area, coastal zone sampling, a lot of the basics are really shared. So we can discuss that a little bit also when I finish. So can you see the entire screen, the presentation in presentation mode, right? So if I start moving,
No,
you see...
just can you make it full screen?
Okay. I don't know how to make that because
on the top right.
Because this is through the... This is very weird because I'm sorry, I'm just connecting on the browser. So when I see the presentation, I cannot see the Zoom. So at the moment, okay, it says it's still sharing, but where is the screen? I don't know what happened to the screen at the moment.
Can you click the icon? Oh no, there's...
Yeah, I stopped sharing, and I'm gonna try it again.
Sure.
Since the presentation should be here, I'm just gonna close a couple other things I had. Because what the selection for entire screen, I don't really see the PowerPoint. So only if I click on the window. And if I go to... Okay.
Can you click that button? The button right next to the volume adjustment.
Where,
the bottom right.
Well, the problem, Dr. Young, is that I cannot see the Zoom screen when I'm doing this.
Oh, okay.
So, wait 1 second. Okay, so Nakshi is trying to help me. So maybe if I put in...
Or can you type F5?
Full screen, maybe like that, and then... You see full screen? Yeah, okay. So in my computer, I see the presentation in full screen. Do you see it now?
Do you have two screens or just one?
Just one.
Oh, okay. Or if you hit function plus F5 at the same time, function key. Do you have a function key? And F5, press F5 at the same time, probably will give you the
I'm not
screen.
Using an Apple computer.
No, it's not Apple. Just in Windows.
Okay. No. Another option is I could try to disconnect entirely and see if I can connect with the app, because the app I've been successful. Would you prefer to do that? I'm sorry, it's taking a bit of time.
That's fine. We'll just wait a little bit.
Okay, so I'll try to be quick coming back.
I'm sorry for some technical issues, so she'll be back soon. So this is a difficulty we have all the time in, online conferencing. So connection issues, some camera issues, and some mic issues. So I'm sorry for that, but please be a little bit more patient.
네, 저 코이카 박소영입니다. 저희 요거 좀 다시 연결될 때까지 한 5분 정도라도 그러면 여기 티 준비되어 있거든요. 가지라고 공지를 하는 게 어떨까요?
뭐가 준비가 됐다고요?
여기 티요.
티슈?
네, 커피랑 아니요. 커피랑 네네 다과가요.
So why don't you take a short break, 5-minute tea breaks until the Dr. Junior is ready. Okay? So go ahead and take a tea, bring your teas and chat each other. Okay? It's a networking time for 5 minutes. And I'll also grab some tea.
안녕하십니까. 코이카 연수사업실장 조형래입니다. 이렇게 영상을 통해 여러분을 만나 뵙게 되어 대단히 반갑습니다. 코이카는 지난해 처음으로 온라인 방식 연수를 도입했습니다. 이를 통해 코로나19로 국가 간 이동이 자유롭지 않은 제약을 극복하고 한국의 개발 경험을 함께 공유할 수 있었습니다. 코로나19의 영향이 여전한 올해에도 디지털 컨택을 통해 여러분들과 다시 만나게 되었습니다. 온라인 교육이 목표한 성과를 거두기 위해서는 무엇보다도 연수생 여러분의 적극적인 참여가 가장 중요합니다. 코이카와 연수기관도 세심하게 준비해서 여러분들이 보람과 즐거움을 함께 얻을 수 있도록 최선을 다하겠습니다. 연수생 여러분, 코이카는 4가지 핵심 가치를 가지고 있습니다. People, Peace, Prosperity, Planet, 즉 4P가 바로 그것입니다. 사람 중심의 평화와 번영을 위한 개발 협력을 실천하고 기후, 환경과 같은 글로벌 이슈 해결에 기여하고자 하며 그 중심에 여러분이 있습니다. 우리 글로벌 연수 사업이 여러분 국가에서 이 네 가지 가치가 실현되는데 기여할 수 있으리라 믿습니다. 여러분은 여러분 나라의 발전을 이끌어 나갈 주역이십니다. 코이카는 여러분의 노력과 열정을 항상 응원하고 여러분이 각자의 역량을 향상시켜 여러분 국가의 번영에 기여할 수 있도록 최선을 다하겠습니다. 마지막으로 코로나19로 인해 많은 어려움이 있으시겠지만 이를 잘 극복해내시기를 바라고 코이카의 연수 프로그램이 여러분께 많은 도움이 되기를 기대합니다. 감사합니다. 안녕하십니까. 코이카 연수사업실장 조형래입니다. 이렇게 영상을 통해 여러분을 만나 뵙게 되어 대단히 반갑습니다. 코이카는 지난해 처음으로 온라인 방식 연수를 도입했습니다. 이를 통해 코로나19로 국가 간 이동이 자유롭지 않은 제약을 극복하고 한국의 개발 경험을 함께 공유할 수 있었습니다. 코로나19의 영향이 여전한 올해에도 디지털 컨택을 통해 여러분들과 다시 만나게 되었습니다. 온라인 교육이 목표한 성과를 거두기 위해서는 무엇보다도 연수생 여러분의 적극적인 참여가 가장 중요합니다. 코이카와 연수기관도 세심하게 준비해서 여러분들이 보람과 즐거움을 함께 얻을 수 있도록 최선을 다하겠습니다. 연수생 여러분, 코이카는 네 가지 핵심 가치를 가지고 있습니다. People, Peace, Prosperity, Planet, 즉 4P가 바로 그것입니다. 사람 중심의 평화와 번영을 위한 개발 협력을 실천하고 기후, 환경과 같은 글로벌 이슈 해결에 기여하고자 하며 그 중심에 여러분이 있습니다. 우리 글로벌 연수 사업이 여러분 국가에서 이 네 가지 가치가 실현되는데 기여할 수 있으리라 믿습니다. 여러분은 여러분 나라의 발전을 이끌어 나갈 주역이십니다. 코이카는 여러분의 노력과 열정을 항상 응원하고 여러분이 각자의 역량을 향상시켜 여러분 국가의 번영에 기여할 수 있도록 최선을 다하겠습니다. 마지막으로 코로나19로 인해 많은 어려움이 있으시겠지만 이를 잘 극복해내시기를 바라고 코이카의 연수 프로그램이 여러분께 많은 도움이 되기를 기대합니다. 감사합니다.
So, Luciana, are you ready to share your screen?
Yes, I think we can try a couple different options. So I
Okay.
actually converted to PDF, and maybe
Okay.
that will help.
Okay. So
It's probably not ideal, but
yeah,
I
well,
can still try what you were saying initially, but I'm not sure.
okay. Why don't you try and see what it can help?
To use this control tab.
So just try to make a full screen and then press Control tab to go back to Zoom, and then you can press the share button on the Zoom menu.
I see. Okay, so let me see. Let me try that. If it doesn't work, I mean, I don't have a lot of animations anyway,
Okay.
so the PDF should be fine.
Yeah, definitely.
And so if I do that and then Control tab, no, it doesn't really.
If you press Control tab, you will see Zoom. No, I'm sorry, Alt tab, not the Control tab, Alt tab.
Okay. Ah, okay, yes. Alt tab, and then I go back to
Zoom.
Zoom. Oh my God, I don't know what is happening. I have too many things. Okay, maybe I should just close the number.
So if you keep pressing Alt Tab, you'll go back to, you'll find the Zoom screen is highlighted, and then go to, and then it makes you control your Zoom menus. Or you can send us file, or then you can
I
press.
actually already sent you the link with
really?
the file.
Although it didn't work. It's like in our institute, the Dropbox link doesn't really work properly because some security issue, they blocked it.
Right. So,
Or
so
you
the
can
email
email me.
didn't go through?
No,
Sorry.
I don't see any presentation material here. I don't see your screen. I just want to see you. So, why don't you
No, what
try
I
to?
was saying is that I sent you an email with the link
Oh.
to the presentation.
Is a Dropbox link?
Yeah, it's a Dropbox link.
Yeah, Dropbox doesn't work here. Our institute blocked the Dropbox link.
I see.
Okay.
So why don't I just go ahead and try
Sure. Okay.
the PDF version?
Sure. Yeah. I think this time is the screen is they're too big, so you can just adjust the size. If you ever press the minus button on top left, yeah, I think that looks good. Luciana, I think your mic is on mute. I cannot hear you. So Luciana, please unmute your mic.
Okay. Can you hear me?
Yes. Yeah, I can hear you. Loud and clear.
And you can see the screen?
Yes. So everyone can see her screen?
Now I cannot hear you, Doctor. Yeah.
Yes, we can see the screen.
Okay. So Luciana, just go ahead. Everyone can see your screen. Okay. So it's good to go.
Okay. Can I go ahead? Is it okay to go ahead?
Yes.
Okay. Okay, so sorry about that. We have lost quite a few minutes, but I'm sure we'll recover it. So after my brief start, so basically what I'm going to go through today is a little or a short background about the ISA, and just some also then talk about the biological communities that are found in the area, or in more particular in the habitats that are currently being considered for mineral exploration. Then we'll discuss sampling and preservation and collaboration opportunities. As Dr. Yang was mentioning, some initiatives that we are developing. And then I'll give or finish with some insights that perhaps can be used for your discussions and your preparations of the action plan. So starting with the background, we'll skip that nice effect. I think I can share then the presentation for you to have it as well. I wanted to just very briefly refer to the mandate that is assigned to the International Seabed Authority by the United Nations Convention on the Law of the Sea. And this mandate can be described in 4 core areas. So one of these areas is, of course, to manage the resource exploration and potential future exploitation activities. And by doing this, ensure the protection of the marine environment and biodiversity. And the ISA is also tasked to promote marine scientific research and share the benefits of the activities, both monetary and non-monetary benefits for the entire humanity. So as you know, it's a very complex and big mandate to put in practice. So for this presentation today, we'll focus primarily on the aspect of the protection of the marine environment. And this responsibility is referred under Article 145 of the United Nations Convention on the Law of the Sea, where it stated that the ISA needs to take the necessary measures to ensure the effective protection of marine environment from harmful effects which may arise from activities in the area. And to this end, the ISA shall adopt appropriate rules, regulations, and procedures. Then this responsibility is reflected in the ISA strategic plan that is currently in action. So from the period 2019 to 2023. And this is one of the 9 strategic directions of this plan. And in particular, for today, we'll discuss aspects that are relevant to the access to environmental information and to monitoring programs and methodologies, all of these that can support the development and the implementation of the regulatory framework, and also to develop the appropriate measures and tools for regional environmental management plans, as well as environmental impact and risk assessment from the exploration activities that are currently taking place. So about the exploration regulations, I refer here to the 3 documents that can be seen on our website and form part of what we call the mining code. And very briefly here, I just wanted to mention that the implementation of these regulations is a collective effort, and so there are responsibilities to different actors in this process. So the Legal and Technical Commission, which is one organ or the advisory organ of ISA to the Council, is responsible for developing and implementing the procedures to assess the potential serious harmful effects from the activities. On the other hand, the contractors who are undertaking the exploration activities should gather environmental baseline data against which the potential impacts can be assessed. And the contractors and the sponsoring states are responsible to cooperate with the authority in implementing programs for environmental monitoring and evaluations. So it's really an effort from many different angles. So now another important document, and I think can be quite useful source of information for other activities and other, in particular for research activities as well in the offshore areas of the ocean, is these recommendations for the guidance of contractors for the assessment of possible environmental impacts. And in this document that was prepared by the Legal and Technical Commission, it describes what and how these environmental baseline studies should contain. Also aspects of data collection and reporting. It mentions the importance of cooperative research and lists the activities that require impact assessment during exploration. So it's not all activities. Most of the activities that are currently being undertaken as part of the exploration phase are very similar to research, to marine research in these habitats. But there's also a list of activities that would require impact assessment, such as testing of component equipment and collection of larger amounts of the minerals, or there's quite a few others. And most importantly is this explanatory commentary, which is a huge part of the document that describes the best available technologies and methodologies to undertake this work. So in terms of environmental baseline studies, these are truly multidisciplinary studies. And as I have highlighted here, the biological communities is one group of sampling parameters, along with many other environmental parameters from physical and chemical oceanography, the geological properties of the sediments and then the substrates, and then a series of other variables that help to understand also ecosystem functioning. This is part of bioturbation, the different nutrient and other chemical fluxes to the sediment, sediment oxygen consumption, and food web structure. These studies are described in an ecosystem approach. So looking at the communities and the different environmental parameters at the seafloor and through the water column and up to the surface, from all ranges of biological sizes and functional groups. And very importantly is that also to describe the variability of the different habitats and this in relation to topography, depth, different characteristics of the sediments, and also the abundance and type of minerals. So for the different, we'll go there a little bit. So there's some differences for the different types of minerals, but it is important that the sampling design is statistically robust and use standardized methodologies and also takes into consideration spatial and temporal variation. So now, finishing with that background, sort of more regulatory framework that we operate with, I'm gonna move into a section of focusing on the biological communities that we find in the area. And just a very brief reminder that the area is the seabed and seafloor and subsoil thereof beyond the limits of national jurisdiction. So we're talking about a little bit over 50% of the ocean floor that is beyond the national jurisdiction. And this is where the mandate of ISA operates, and where we have at the moment issued 31 contracts for 3 types of mineral resources. So this map shows that most of the contracts are happening here in the Central Pacific Ocean, in a region called the Clarion-Clipperton Zone, where we find polymetallic nodules. And there's more recent contracts that have been issued for the Northwest Pacific region, and this is for the cobalt-rich ferromanganese crusts. And so there's 4 for crusts and 1 for nodules here. And then we have polymetallic sulfide exploration, both in the Indian Ocean and in the Mid-Atlantic Ridge, and another contract of nodules also in the Indian Ocean. And the contract here in the South Atlantic in Brazil is currently being withdrawn from ISA, as this is located in the claim for the extended continental shelf. So there's been some work there, but it will be in the future managed by the Brazilian government. So now I'm going to give just a brief overview in terms of what we know currently of the biodiversity across these different types of habitats. And you may know that polymetallic nodules, which are those black dots in the images here, they are found on top of the sediments and in relatively flat areas of the ocean floor called abyssal plains, and they lay between 4,000 to 6,000 meters depth. Like I said, there's currently exploration for polymetallic nodules in the Pacific and in the Indian Ocean. This habitat is relatively, and as we get more knowledge, we're starting to understand more of the heterogeneity in these habitats. But in terms of temperature, is a relatively stable habitat to 2 to 4 degrees Celsius, and is typically with very low food. So the most of the food input for these organisms comes from the surface through so general the surface productivity and the fall through the water column, and occasionally from some larger inputs from whale carcasses or other large fish and mammals that can, when they die and they sink on the seafloor. But in general, there's relatively low availability of organic matter. And we know in terms of biological communities that there's a high diversity, but relatively low abundance. So most of the animals have very sparse distributions, and they occur in very low numbers. There's typically mobile scavenging communities. So there's fish and octopus and shrimps especially in that benthic boundary layer, so a few meters above the seafloor. There's also many different types of suspension feeders that can live attached to the nodules or to a few other hard substrates, So occasionally there are some seamounts as well, but typically the nodules are in the flat areas, not on the seamounts. And they can have different kinds of fauna attached to them. And then there's a wide range of animals that live inside the sediments, many worms, some mollusks, and many other echinoderms and so on. So this habitat is very, very contrasting with the polymetallic or the areas where polymetallic sulfides are found and are currently being studied and explored. These are located in the mid-ocean ridges associated with hydrothermal venting systems. And the range of depths that these type of mineral occurs is much broader. So it could be from less than 1,000 to more than 5,000 meters. So much wider depth range. Like I said, currently there's contracts. There are in the mid-Atlantic region in the Indian Ocean, and these habitats are typically characterized by very heterogenic and very variable temperature. So if you're looking at the black smokers, like I'm pointing here, these in the chimneys, these fluids can have up to 400 degrees Celsius, but then a few just centimeters, a few meters outside these chimneys, again, we are within 2 degrees Celsius. So there's the animals that live around these areas have developed a very extreme tolerance to these changes in temperature gradient and also chemical gradients, and therefore are very are well adapted to these environments, and in most cases are endemic species only found in these areas. Here the food comes mostly from the chemosynthesis that is based on the chemical reactions from the methane and the sulfide that comes out of the chimneys. There's also another part of the food web that is or that relies on the surface productivity as well. And because of all these habitat changes that I mentioned, one key aspect is that in these cases we have less or the diversity is relatively low, but we have very high numbers for each species. So there's very high abundances. There's dense mats of sessile chemosynthetic animals like mussels and tube worms, and there's lots of shrimp typically also living in this or feeding from the fluids. There's bacterial mats and many types of suspension feeders and grazers. And then the third type of mineral that is being explored at the moment, we could say that it falls a little bit in between those two that I said. So in terms of heterogeneity and in terms of biological diversity. So the cobalt-rich ferromanganese crusts are typically found in seamount features. So typically more flat seamounts and around the top of that. And again, the depth range is not as wide as for the sulfides, and a bit and shallower, and typically shallower, from less than a meter to 2,500 meters. We currently, well, there's exploration in the Northwest Pacific and the South Atlantic. Again, and temperature-wise, it's more stable, and it's a place typically very productive, or areas that are very productive in general, and this has to do with some of the current systems and the trapping of organic matter around these geological features, and typically where many fish aggregate, and there's also various types of coral and sponge gardens associated. Again, in terms of abundance, this is medium to high compared to the previous habitats that I described. So this is, in general, the biological communities that are being considered or studied in more detail with the work of the contractors from ISA. And when we now focus in terms of sampling, the recommendation, again, like I mentioned before, is to describe the communities as a whole. So looking from the microbial communities, all different types of microorganisms that are very, very small, to then the meiofauna, which is typically described as being larger than 32 micrometers, but less than 300 micrometers, where we then start to define macrofauna. And then the megafauna, which is bigger than 2 centimeters, and we can see it usually with videos, and it's more human visible without microscopes. So in terms of sampling, again, I'm going to refer to a series of tools that are now being used for this work. This may not necessarily be used in shallower habitats, but the principles are I think, are very similar. So in terms of looking at, or what we need to consider in terms of being able to characterize the biological communities in terms of abundance, taxonomic composition, diversity, and the different trophic groups. So what is commonly or what should be used for microbial and meiofauna communities is what we call these multicorers. So there's a series of tubes that collect sediment, and then we can study the sediment layers. So we can cut these cores into different layers and study from the close to the surface, or the interface between the water and the sediment, and then down a few centimeters. And then the box corer, it's larger, well, typically 50 by 50 centimeters box that goes on the sediment, and then collects that big cube of sediment, and then we can study again in layers. And then for example, for the fishes and scavengers that are swimming around close to the sediment, there can be different kinds of camera systems that can be used to study them. And this is an example of a fixed camera, so that could do time-lapse recording, so with time, over time. There are other kinds of camera systems that can move more freely and be used for studying larger organisms, like I was saying. So the two cameras and the remotely operated vehicles. And the difference is that the remote operated vehicles have the mechanical arms that can collect specimens and put them in the boxes here to bring them to the surface, whereas this camera is a simple camera system, so only records video. And then, as I mentioned, we also need to look into the communities that are found in the water column and at the different layers. So this, for example, a multi-net system is a series of plankton nets that can be deployed from a vessel and then at the different water depths being triggered, so we can sample separate portions of the water column. And there are other types of instruments that are being developed. For example, this one, that these tubes here function as plankton nets, and they are attached to a vehicle that can just move around, and we can control better where it goes, and it collects the samples, and it really can go very close to the bottom, while, for example, this system of nets cannot sample the 50-meter layer closest to the sediment because of technical constraints. So very importantly is that in terms of describing the community, the biological communities, it's also important to understand other aspects of the environmental parameters and the conditions that drive these communities. And there's here different types of equipments that are used to describe the seafloor topography, as well as collect water samples with this water sampling carousel for all kinds of chemical and oxygen and temperature, salinity, and so on, various parameters of the water. And then I mentioned before the multicorer for collecting sediment. This can also be used to study the characteristic, the geological properties of these sediments. There's different kinds of corers. This one is used with the push corer is used with the remote operated vehicle. There's sediment traps as well to collect information on the fluxes of the sediment, and again, some of the more advanced underwater autonomous vehicles that can also do some mapping and collect video, and in some cases collect also some have some sensors for water properties. So, like I said, these refer to those other variables that can be helpful to describe the environment. Again, as I said, these differences in the different in the various equipments, they take into account their usefulness to describe some parts of this variability. So for larger animals, you use larger equipment and so forth, and the number of samples that are needed also vary for each. And then in terms, the other aspects that are also important is to look at more aspects of the ecosystem functioning and connectivity. And there's also a number of, so the document that I referred also lists different methodologies used to do this, including toxicology to look at the animal tissues, and other aspects of migration and exchange of genes for connectivity purposes. So sorry, not really seeing the here, I think that's where I had a bit of animation. So the final slide is a little bit crowded, and I'll try to go a bit slower on this one because I think it's the last one referring to this the main topic of this lecture. And I've already went through most of the aspects of the collection. So this idea or the message that is important is the need to use different and multiple sampling gears to collect not only the biological specimens, but many other aspects of the ecosystem, that to be able to understand the patterns of biodiversity. And then, as I said, it's also, or the immediate step after that, and in many cases, because we have these mixed animals and sediments, and we have, or with water, we need to first sort them, or at least be able to separate them by sizes, and we can better study them afterwards. So this part of sorting can be initiated on board of the vessels, or but then in most cases is finalized once we are back in the lab inland. Mostly is done on board, more for conditioning of the samples and being able to preserve them properly and to transport them. In some cases, or ideally, we could do more sorting already and already pick good samples and good specimens for better preservation and also for the identification. So in terms of preservation, it again really depends on what's the purpose of the study, and preferably we would preserve samples that can be both used for DNA studies or extracting the DNA afterwards, and alcohol is the selected preservative, but there are others that can also be better to keep the morphological characteristics of the animals, and or for other kinds of studies if we need to do more toxicological or isotope analysis in the tissues. There's also different protocols for that, and again, more details and references are found in that document with the explanatory commentary that has been prepared and compiled by the Legal and Technical Commission. So I'm not going to go much into the next three items, the identification, archiving, and data use and sharing, which are going to be addressed in the following or more in the following lectures, but I wanted to put them here as to show the entire data flow or data workflow when we, from the collection to the archiving and using of the data, which is in the end what we all intend to do. Yeah, so we can discuss some of these, but I would prefer to move on, especially because I'm seeing also the time going, and we can leave some time for some discussion at the end. So to finalize, I'm going to try to go a little bit faster in this last part, but I'm happy to then share more through email if needed. We can continue this discussion. So it's now referring to the collaboration opportunities, and these are being developed or highlighted in ISA through its mandate on marine scientific research to promote that research in the area and coordinate and disseminate the results of this research and analysis. So in this context, ISA has, the Assembly of ISA has adopted an action plan for marine scientific research in support of the United Nations Decade of Ocean Science, and which is aligned with ISA strategic plan, a high-level action plan, and the mandate from UNCLOS and the 1994 agreement. In this document, there has been identified 6 strategic research priorities, and for today I'm focusing primarily on the work that we are doing on the priority number 2 to standardize methodologies for biodiversity assessments. I'm happy to share then through Dr. Yan the documents and as well as the recent reports on the activities that are being undertaken. So one core aspect of this research and collaboration opportunities is through our database system called DAY2. And DAY2 is where we collect all the information related to the activities, related both to the resource assessment aspects and the environmental studies. So yeah, here you see the type of information that is associated with deep data and that is publicly available is the environmental data. So all environmental, including biological data, is made available to the scientific community and the general public. There's also data that is, so the contractors should report data using the digital templates. These are also available in our website, but they also report and send other types of data and information associated with their studies. So I have not updated these slides, unfortunately, for the 2021 cruises, which adds a few more here, but this is just to show you the range of or the number of cruises that have already taken place in the area in the past 20 years. For polymetallic nodule exploration is over 100 cruises. And for sulfides and crusts is less, as the regulations were also adopted later, or about 10 years later. And but the numbers continue also to rise. So there's a lot of information being compiled and uploaded into deep data. And since it was launched in 2019, it has increasingly collected all this information. And from earlier on, as exploration started, there's been a number of activities related to the taxonomic standardization. So there were some earlier studies publishing protocols for the different types of fauna and different organisms. There's been a series of workshops to compare and calibrate identifications. And there was also a synthesis workshop where all the information for the Clarion-Clipperton zone was compiled, and this was also to support the review of the environmental management plan that is in place for this region. And in the past 2 years, we have been very much appreciated and grateful for the collaboration of MABIC and the Ministry of Oceans and Fisheries from Korea to organize online workshops. The first one in 2021, where we discussed really different approaches that can be developed for enhancing collaboration and improving taxonomic standardization. Last year, we focused, or was the follow-up workshop was focused on image-based assessment. So the increasing number of images and videos that are being taken in these areas and how they are being used and developed as a more cost-effective and also less invasive, less destructive technique to study these habitats. But there's still a lot of underlying work to allow these techniques to be done. And so we discussed, the reports from these workshops are all available in ISA website. And from these workshops, we drafted a roadmap to facilitate integration of taxonomic knowledge into ISA's efforts for protection of marine environment. I'm gonna skip through some of these slides as I see the time running, but basically this roadmap focus or has highlighted different activities that needed to be done more short-term and long-term to promote or to enhance taxonomic standardization, and these fall into aspects related to data sharing, to the data quality, and the different methodologies or standardization of methodologies for sampling. And across all the workshops, there are always two very important topics on capacity building and awareness raising in relation to taxonomic work. So in terms of exchanging data and information, we've started also to implement some of these suggested activities in terms of interlinking deep data with other global databases, so the Ocean Biodiversity Information and the WORMS, World Register of Marine Species. So we established collaborations with these two entities so that our data is now also part of the global data system, for marine for ocean biodiversity. And there's a number of additional things that we still need to do and develop, some toolkits, but I can mention that a bit later. Again, some activities that need to take place to ensure quality and control of the data, and the aspects, again, relation to those intercalibration exercises, the importance of preserving and keeping long-term voucher specimens, the importance of having good metadata associated with all the information. And as I said, capacity building is a very important aspect that we are focusing on and will continue to work. So on this website of ISA, there's a number of existing ways of doing training, either through internships, the contractors training programs, and there's a number of other initiatives also starting to be developed for further training. And again, what we want is to try to assist in making all the data and the services related to taxonomy more accessible and used by wider community. So I'm going to conclude quickly with the Sustainable Seabed Knowledge Initiative that Dr. Young mentioned very briefly. And so this is an initiative that we launched this year, but it's really how we're going to put in practice all those activities that I mentioned that are part of our roadmap. So again, I don't need to repeat, but there's 5 key components of this initiative that we'll be working on in the coming years, from improving the knowledge and describing more species, but developing tools, promoting data sharing, again, capacity and opportunities for networking and outreach. Here is a brief summary of the recent review that took place for the existing data and deep data database, as well as other literature and public sources. And this number keeps growing. At the moment, we have a list of 653 benthic species. This excludes microbes. And you can see the number of descriptions and the number of publications over the past 5 years has increased exponentially. So 30% of those species were described in the last 20 years. And in 1 year alone, these 66 species were described. So we really want, with this new initiative, to keep increasing these rates and improve the knowledge of the biodiversity for many of these areas that we have been studying for many years. So to finish, I want to reinforce the announcements for the upcoming workshops. So there's the Inception workshop for the SKI initiative that will take place in Mabic. So I'm very happy to finally visit Mabic after these few years of collaboration. And before this one, we'll also have the ISA Mabic, the third workshop, and this will focus on enhancing genetic approaches. So we focus on images last year, and we'll focus on molecular data and information this year. So this was mostly what I had to say. I'm really sorry for the time I spent with the technical glitches. I had some insights for the action plan, which I'm happy to share as well. But I basically wanted to just, for example, give some idea, maybe here as an example, how you could incorporate or think about the challenges that you are needed to address, and who and how can assist in overcoming those challenges. And again, taking into consideration gender equity, which is what this workshop is about. So I'm sorry I rushed a little bit. I don't want to take time from my colleague in the next lecture. And I'm happy to share my slides and also answer more questions if you have them afterwards. Thank you.
Thank you, Luciana, sharing your expertise on biological sampling, especially in deep sea. Can you hear me?
I cannot hear.
Okay, can you hear me now? No? All right. I think Luciana is having connection issues or speaker issues. Is anyone else can hear me? Raise your hand or just, okay, so.
Is it only me? I can hear, you can hear me, but I cannot hear you.
Okay, I can hear you, but
okay.
You can hear. All right, so. Okay, there is something going on between, just, I don't really know. I just can't understand why I can hear you, but you cannot hear me.
Okay, so what's going on?
Can you turn the volume up, your speaker?
Should I just leave again and come back?
No. So while Luciana is ready, anyway, thank you, Luciana, sharing an expertise on sampling biodiversity. So you can hear me now?
Yes.
Okay, all right. I saw a lot of interesting pictures of unique environment and organism, as well as really cool equipment, which is really challenging environment. I really enjoyed your lecture. Also, thank you for announcing ski, which will be held in Mavic in November, and also online. So I hope the participant today may join us online if possible. It is open to public, or just open to only the international?
Yeah, for ski inception workshop, we will have a more limited
I see. Okay.
participation for technical discussion, but then on the third day it's open to everyone. So you can register for that link on the for the public session.
Okay.
But for the other workshop, it's also trying to be in person, but I think we may open also the last day. I think it would be important because I'm afraid there will be some difficulties in reaching or some participants to travel. Anyway, there's some funds available to support in-person participation. So I wanted also to highlight that in case there are interested people here from Fiji. So again, the registration is open. This one closes soon, so I really encourage you to go through the websites and or ultimately contact me or Dr. Yang.
Okay. So
if you have any questions,
if anyone here you're interested in to come to Korea, or just join it online, just let me know and I'll give further information later. Okay. Now we will have discussion on this biological sampling. Again, Dr. Luciana facilitates this session. And dear participants, if you have any question, please press response button on the menu at the bottom of Zoom app and press hand up. I don't know, it's like my app is in Korean, so I have to even translate the menu button. So hand up so we can see who raised the questions. Okay, so raise your hand on the app. And also so Luciana hadn't had much time discussing about the action plan. And this is a good time. You can ask action plan regarding deep sea activity or action plan in general. She's much better than me, in terms of action plan. So if you have a question, raise your hand. No one has, so we'll wait a bit.
Yeah, maybe give a few minutes for for thinking. But I think what I wanted to mention again, and I said that in the beginning, and I know that some of these, or in particularly the sampling methods that I was referring to are quite specific for deep ocean and offshore waters. And I think that this might be not necessarily what is used and available for the work of most of you. But the idea here is really to highlight more of the importance of collecting not only the biological specimens, but other aspects of the environment that help us understand the biological specimens. And I know I've seen some country reports, and I know there are different challenges that are more at the data collection and the data management. Also some challenges with the taxonomic identifications, but which I think we can really discuss across, whether you sample a specimen from or a fish that is found offshore or a fish that is collected and closer. So there's definitely key aspects that are transferable in terms of improving the expertise available for that. And in terms of the sampling, I saw also some challenges referring to the preservation and sometimes the difficulties of preserving the specimens on the vessels or on the different boats or whatever where those samples are collected, which is also a very critical aspect that happens when we're in offshore and sometimes running out of alcohol, and we face different issues, and not all the cruises are equipped with cold rooms or with freezers and fridges ideally. So a lot of what I showed is really quite advanced available technology and available methods. But I think there's more and more discussion on finding alternatives, and there's some, I think, developing kits and other like sort of low cost tools that can really replace some of these tools for different purposes. And I know that of a small camera that is being developed that can more easily be assembled and used even from a small vessel that doesn't need like the big ocean going cruise. So I wanted to sort of spark a little bit that aspect, and then I'll be happy to then perhaps through Dr. Young's share a bit more if some questions come during the next coming days. Okay. You can share my email as well, or combine questions and send me, so we can see how best to incorporate that if possible. It's unfortunately that we have some conflicting events towards the end of the week, so which may actually not happen, Dr. Young, because there's a storm. So we are now going through a big storm, tropical storm here. So we'll see. In case I'm more free towards the end of the week, I'll let you know, and I can join the next coming days. Yes.
All right, so I'll just wait just a little bit, Luciana. This is, even though we have a gender mapping section tomorrow, if you guys have any question in gender equity or gender balance in this field, this is a good time to ask a question one last time. All right. So I'll have Luciana here for 5 minutes, another 5 minutes. If you don't have any question, I'll let her go and sleep. Okay.
Thank you.
All right. Oh, seems like the participant this time is a little bit shy, and okay, so
they're still warming up. It was a lot of information. It was a lot of information.
I don't want to receive a question next session because I'm not an expert in gender equity, and so if I have a question on this matter,
for that, my colleague will join you tomorrow.
Yeah, tomorrow. The Sonakshi is the right person you can ask.
Yes.
All right. So seems like there is no other question. So I'll let Dr. Luciana go. So why don't you give a big round of applause to Dr. Luciana? Thank you, Luciana. All right.
Thank you very much, and I really hope to see you or some of you in Korea, maybe.
Okay.
Towards the end of the year. So thank you very much, and all the best for your discussions.
Thank you for your support. All right. Thank you.
Thank you.
So this is about 2:37. So why don't we have 10 minutes break? 30 minutes break, and we'll come back 2:50. Is that okay?
Okay.
All right.
Okay.
See you in 30 minutes.
Okay, fine.
Hello everyone. In this video, instructions on how to complete the pre- and post-surveys on fellowship programs will be provided. Kuika kindly asks you to participate in the pre- and post-surveys. The purpose of these surveys is not to measure the level of your knowledge, but to assess the effectiveness of the fellowship programs. The accomplishment level is measured by comparing the pre- and post responses, thereby directly affecting the management of program outcomes. Your cooperation will be much appreciated, as your feedback is greatly helpful to improve the quality of the fellowship programs. Now, I will walk you through how to complete the survey. There are two questionnaires, the pre-questionnaire and post-questionnaire. Please complete the pre-questionnaire at the beginning of the program and post-questionnaire at the end of the program. The purpose of conducting the same surveys twice, before and after the program, is to see if there is an increase in accomplishment level with significant difference after the program. The same questions on learning contents of A, B, and C are asked under the three categories, which are cognitive knowledge, technical skill, and attitude. Here, learning contents A, B, and C are the three must-to-know topics that the training institutes expect participants to learn at the end of the program. Please read all the questions carefully and mark the relevant response among options 1 to 5. All responses are on a 5-point scale, with 1 being the lowest and 5 being the highest. For instance, you might have a relatively low level of prior knowledge or experience, falling into option 1 to 3 in the pre-questionnaire at the beginning of the program. But that does not mean that you are less qualified or subjected to disadvantage. At the end of the program, if your level of cognitive knowledge, technical skills, and attitude aspects have improved, you can mark option 3 to 5, indicating a relatively high level of competence. Given that the surveys are based on self-assessment, your honest and objective responses would be appreciated. Seeing is believing. Here I will show you how to participate in surveys through Google Form. The following page will appear when you click the link for the survey. You need to log into your Google account to start the survey. Please check the box below after watching this video, and make sure to answer all the questions. All questions must be completed without blanks before you move to the next page. Please enter your first name and then last name. If you select Other, please enter your agency type directly. Please enter only numbers. Please enter 8 digits. The survey measures the cognitive knowledge, technical skills, and attitude aspects of each learning content A, B, and C. 1 is the lowest, and 5 is the highest. Please check the number that describes your current level of cognitive knowledge, technical skill, and attitude. Now, here's a notice regarding copyright for online fellowship program. Copyright in the course materials of the online fellowship program belongs to COICA and lecturers and is protected by copyright law. Participants must comply with copyright laws, and the following acts are prohibited. First, lecture materials such as PowerPoint files, videos, etc. are open to participants for training purpose only, and commercial use is not allowed. Second, video recording, capturing, and audio recording of lectures or sharing them with third parties are prohibited. Third, distribution and reproduction of lecture materials without the explicit prior consent of the copyright holder is prohibited. Lastly, sharing or handing over of ID and password of the lecture platform to a third party is prohibited. Participants are solely responsible for any disadvantages that may occur in the event of a violation of copyright law. For successful online fellowship program, your active participation is needed above all. Now, please check the box below and continue to the next page. Thank you. Hello everyone. In this video, instructions on how to complete the pre- and post-surveys on fellowship programs will be provided. Kuika kindly asks you to participate in the pre- and post-surveys. The purpose of these surveys is not to measure the level of your knowledge, but to assess the effectiveness of the fellowship programs. The accomplishment level is measured by comparing the pre- and post responses, thereby directly affecting the management of program outcomes. Your cooperation will be much appreciated, as your feedback is greatly helpful to improve the quality of the fellowship programs. Now, I will walk you through how to complete the survey. There are two questionnaires, the pre-questionnaire and post-questionnaire. Please complete the pre-questionnaire at the beginning of the program and post-questionnaire at the end of the program. The purpose of conducting the same surveys twice, before and after the program, is to see if there is an increase in accomplishment level with significant difference after the program. The same questions on learning contents of A, B, and C are asked under the three categories, which are cognitive knowledge, technical skill, and attitude. Here, learning contents A, B, and C are the three must-to-know topics that the training institutes expect participants to learn at the end of the program. Please read all the questions carefully and mark the relevant response among options 1 to 5. All responses are on a 5-point scale, with 1 being the lowest and 5 being the highest. For instance, you might have a relatively low level of prior knowledge or experience, falling into option 1 to 3 in the pre-questionnaire at the beginning of the program. But that does not mean that you are less qualified or subjected to disadvantage. At the end of the program, if your level of cognitive knowledge, technical skills, and attitude aspects have improved, you can mark option 3 to 5, indicating a relatively high level of competence. Given that the surveys are based on self-assessment, your honest and objective responses would be appreciated. Seeing is believing. Here I will show you how to participate in surveys through Google Form. The following page will appear when you click the link for the survey. You need to log into your Google account to start the survey. Please check the box below after watching this video, and make sure to answer all the questions. All questions must be completed without blanks before you move to the next page. Please enter your first name and then last name. If you select Other, please enter your agency type directly. Please enter only numbers. Please enter 8 digits. The survey measures the cognitive knowledge, technical skills, and attitude aspects of each learning content A, B, and C. 1 is the lowest, and 5 is the highest. Please check the number that describes your current level of cognitive knowledge, technical skill, and attitude. Now, here's a notice regarding copyright for online fellowship program. Copyright in the course materials of the online fellowship program belongs to COICA and lecturers and is protected by copyright law. Participants must comply with copyright laws, and the following acts are prohibited. First, lecture materials such as PowerPoint files, videos, etc. are open to participants for training purpose only, and commercial use is not allowed. Second, video recording, capturing, and audio recording of lectures or sharing them with third parties are prohibited. Third, distribution and reproduction of lecture materials without the explicit prior consent of the copyright holder is prohibited. Lastly, sharing or handing over of ID and password of the lecture platform to a third party is prohibited. Participants are solely responsible for any disadvantages that may occur in the event of a violation of copyright law. For successful online fellowship program, your active participation is needed above all. Now, please check the box below and continue to the next page. Thank you. Hello everyone. In this video, instructions on how to complete the pre- and post surveys on fellowship programs will be provided. COICA kindly asks you to participate in the pre- and post surveys. The purpose of these surveys is not to measure the level of your knowledge, but to assess the effectiveness of the fellowship programs. The accomplishment level is measured by comparing the pre- and post responses, thereby directly affecting the management of program outcomes. Your cooperation will be much appreciated, as your feedback is greatly helpful to improve the quality of the fellowship programs. Now, I will walk you through how to complete the survey. There are 2 questionnaires, the pre-questionnaire and post-questionnaire. Please complete the pre-questionnaire at the beginning of the program and post-questionnaire at the end of the program. The purpose of conducting the same surveys twice, before and after the program, is to see if there is an increase in accomplishment level with significant difference after the program. The same questions on learning contents of A, B, and C are asked under the 3 categories, which are cognitive knowledge, technical skill, and attitude. Here, learning contents A, B, and C are the 3 must-to-know topics that the training institutes expect participants to learn at the end of the program. Please read all the questions carefully and mark the relevant response among options 1 to 5.
So welcome back, everyone. Is everyone on the line? Okay, so we'll move on to the next session. So next the next topic is the taxonomy, and for this lecture, I will invite Dr. Beck. Dr. Beck is right next to me. All right, so. So I just want to know that all this presentation material is prepared by Dr. Beck. And let me share the presentation material first. So Is everybody can everybody see this slide? Okay, so Dr. Beck's name is right bottom of the corner, right bottom of the corner. He's prepared all these presentation materials, and he's one of the leading scientists in Mavic in terms of taxonomy, and he asked me to interpret, so I'll give a presentation on his behalf. Okay. Today's topic is about taxonomy, which is the first step in study of living organisms. So the presentation have 4 parts. The first, we'll be discussing about the definition of taxonomy, then the basic unit of taxonomy, that is species. In the second part, I'll talk about the identification and how to collect the sample from sea and how to transport them to the laboratory and how to identify them. And the third section is describing how taxonomies and species when that is unknown or to be known to other researchers. In the final session, there's a summary of the presentation. Okay, Dr. Beck said, you cannot learn everything about taxonomy from this single presentation, since taxonomy is a very sophisticated area of study, and being a taxonomist requires years of training and practices. But I hope by the end of this presentation, you will learn some basic idea about what taxonomy is and why we do perform taxonomy. Okay, let's move on. In this slide, I would like to define what taxonomy is. As you see, the term taxonomy is combined word of taxi, meaning arrangement, and nomos, meaning law. So the term taxonomy can be understood as a law of arrangement. So taxonomy refers primarily to the series of actions, such as naming an organism, characterizing its characteristics, and preserving it as specimen. As you see the right side of the table, there's another term called systematics. That is similar to taxonomy. While taxonomy is interested in single species, systematic can be thought as focusing the evolution of species and the relationship between species. As I briefly mentioned earlier, the basic unit of taxonomy is species. However, the researchers have often different view on the definition of species. Some define it as most basic unit, which is biospecies, meaning biological species. And another scientist called it as ecospecies, meaning ecological species. While some others define it as morphospecies, meaning morphological species. Definition is here, it's quite complex. So just read it through. So it's a biological aspect, ecological aspect, morphological aspect. Let's move on. However, there are many organisms on Earth that cannot be described by single definition. For these reasons, taxonomists often classify species using several species definitions at the same time. So in general, morphospecies, which means like a morphological characteristic, is the most widely used, and ecospecies with biogeographic information, and the concept of evolutionary species considering gene flow are also used additionally. This is kind of quite popular diagram of hierarchy of classification, right? I think almost everybody knows this. Taxonomies use 7 ranks, as you may know, from kingdom to phylum. class, order, family, genus, and species. The hierarchical grouping is called taxon. In taxonomy, an organism is classified into 7 levels according to its morphological similarity. Now, let's see how to define a species. Again, that is a basic unit of taxonomy. It is fair to say that a species is defined as a specimen in taxonomy. Thus, the most representative specimen, including a species, is called holotype. Holotype is unique specimen designated to fix the linear name of a species. And there is a similar term called paratype, where each specimen of type series is other than holotype. When a taxonomist discovers a new species, she must sample the individuals that best reflect the characteristic of the species and designate it as holotype. In addition, she must limit the range of species to paratype when they are judged to be of the same species as holotype. Did you get that? Okay. In the current slide, show that the 2 species A and B can be clearly distinguished. However, in reality, it is almost impossible to collect a paratype that reflect all the variation of the characteristic of the species. Let's get to more realistic example. Here, 2 species A and B are not clearly distinguished, as in the previous slide. There is also chances that taxonomists may designate a sample as holotype, which may not best reflect the characteristic of species. In particular, as shown in the figure, if a species having both characteristics of species A and B is designated as a holotype, it will impact the follow-up studies as well as the species identification. And the next scenario is worse than the previous one. Sorry about that. Okay. Here, the species A have limited sample. There are not many samples and overlap with both holotype and paratype from species B. These species are usually described as cryptic species or sibling species. These species can be classified further into 2 or 3 new species by taxonomists based on another additional information, such as genetic information, chemical information, or biogeographic information, or combine everything. And this is probably the most realistic example. Researchers, including Dr. Beck, often collect samples shown on this slide. The sample contains 3 species, and their characteristics may be very similar. So it is very likely that even in experience, it is very likely that inexperienced researcher will report all 3 species as the same species, which is not correct. To avoid this, today's taxonomists carefully define species by considering genetic information, biogeographic information, as well as, of course, morphological characteristics. So life in nature has wide range of variation. So identification of organism is not really easy task for even a seasoned taxonomist who has been practicing for decades. Therefore, the capacity building in this field is not quite easy. To become a good taxonomist, countless lessons and advice from experienced seniors, a lot of patience and practice are essential. Okay, the next section is taxonomic identification. So we are going through describe how we will actually collect organisms and conduct taxonomy studies. I will mainly show you how to study benthic organism from this from the shore. Okay. All right. Yes. To collect samples, yeah, we dive first. And these short videos show our Mavic scientists collecting samples in Palau. So as you seen in this slide, on the bottom of the slide, that the organism pictures, each organism has different characteristics. So we rely much on experienced divers for collecting samples. And for the next slide, it shows the sample collection activity on collecting boat. There are places when diver cannot access, such as areas of dangerous marine life, like sharks, or deeper, very deep water, like deep sea area in the previous presentation, or very high turbidity. That's really, really difficult term. So for example, unlike very clear ocean in Fiji, the yellows in Korea is very murky, and the underwater visibility is about 0.1 meter. Can you imagine that? You guys have a really, really clear and nice water, but here in some area in Korea, it's really, really dark and murky. In this case, samples are collected from the ship using such as grab. You can see in the slide. Because mainly sediment is collected, the sample is fixed on the boat first and analyze the sample in the laboratory later. This short video shows a team of Mavic researchers, including Dr. Beck, conducting a marine biodiversity survey on the coastal area. Shall we watch the clip? Okay. So what I have to do after once we get some sample collection? I want to tell you that this is not a video with film ourselves, but I brought this video to show how the microscopic image is obtained from algorithm collected from sampling bot. And this video can be viewed at the address below. And if you search on myofauna in the YouTube, you can find many related videos. Okay. These are the organisms that pass through a safe mesh size of 1,000 micrometers as upper limit and are retained on a safe mesh size of 45 micrometer or 63 micrometer as a lower limit. So very tiny organism. And this is what you can see in the sediments from an ocean before fixation. This is not visible with your naked eye, so we use a microscope. And this one shows how we proceed the sample from the sediment in the laboratory. So in terms of myofauna, we need to separate the organism from sediments. So we rinse or color to make it easier to detect. In order to sample fixation, we can use either alcohol or formaldehyde for fixation. Although formaldehyde is excellent choice for sample fixation, alcohol is currently preferred for later sample utilization such as DNA sequencing, and also to avoid toxicity. Formaldehyde is known as toxicity. Okay, I'll just let these video clips go on.
It's not a means you utilize to get somewhere. Somehow didn't notice friendship is an end. What do you know? It happened again. How come no one told me all throughout history? The loneliest people were the ones who always spoke the truth, the ones who made a difference by withstanding indifference. I guess it's up to me now. Should I take that risk or just smile?
Okay. Once rinsing and centrifugation, and if you clearly sort this up, you'll get the organisms collected. You'll get organism like shown like this figure. Then classifying organism at this species, then we need to specify what these are. And classifying organism and species levels requires closer observation using various taxonomic methods. So you can see the nematoda and many other amphipoda and many other small living organisms. And then we have to classify, identify what they are. And in this slide, the classification of organism you see in the current slide is a class level or order level. In Korea, the level of classification in family or class or order level can be done by someone with a college level taxonomic education. And more level of classification, such as family, genus, species, requires specialized references and knowledge of each taxon. So from this level on, the level of knowledge revealed about organism vary greatly from taxon to taxon. Some taxa are well organized and to make it easy to find a genus or even species in a book, while others have really literally little information about it. Some species there is no evidence or no information is known. In case of the order Harpyticoida, which Dr. Beck is researching, researchers have systematically organized information on that taxa. So it's relatively easy to understand and study. And this one shows the researchers try to find the taxa based on the reference, which is they're looking for a similar organism based on the pictures in reference. And here, this one shows the classification keys such as 1 and 2 in the figure. So based on this classification V, this classification V is the foundation of looking for our species or taxonomy levels, determine the taxonomy level. And this slide shows the organism called Tegastidi, which is obtained by our researchers. So this one looks similar to the picture we just saw on the slide before. Do you see the similarity between them? This one and this one? If you have a good eye, and you definitely can tell that is quite similar. For me, I'm not a trained taxonomist, so it's kind of not quite clear. But anyway, yes, Tegastidi. And now let's go down to our genus or species level. The previous slide is in the family level. So this one shows the level of identification process to looking for genus and species level. If your sample meet the first criteria, which in P2, P3 export 3 segmented, then you go to the item 2. And if this evidence met, which is P2, P3 and adopts 2 segmented, this is fourth line from the top, then you can tell that your sample is Arawella. Okay, this is how taxonomists determine the species and genus. Okay. Let's move on. Okay. This ensures that some cases in some sample characters is not exactly the same as those of an existing species. And yeah, some cases just exactly the same species, some cases are not. For close observation, taxonomists occasionally describe the characteristic by drawing image they see under their microscope at the same time. So here, Dr. Beck drawing a sample while he's looking through the microscope at the same time. Fascinating, isn't it?
Stories. We think this is plausible, but it's really hard to get experimental evidence for any of that. I think people are more familiar with some of the offensive.
Again, he's drawing this image,
for example,
looking through microscope at the same time. So you need some lots of training for this and practice for this. So sometimes organisms are dissected into 5 to 7 parts based on the body parts for more detailed observation, because sometimes it's not easy to identify them as a species or genus level. The characteristic of species can be clearly identified after such careful observation. Typically, these observations are made with a microscope capable of magnitude of 600 or 1,000 times. So high resolution microscope is necessary. Okay, moving on. So once we identify the taxonomy, so this is time to, and let me give the overview of what researchers do when this observed species is considered as a new species and how we describe them. So yeah, so of course, as I explained before, the scientists draw morphological characteristics, as you've seen the video before. And there may be some structures are so difficult to observe even when viewed at 1,000 times magnification. In that case, a more detailed structure can be observed through an electron microscope as to better observation. So once you figure out all the characteristics, the observed features are described in detail in writing so that other researchers can easily understand what species are, what these species are. In addition, it is desirable to write it down with the classification key so that other researchers can easily find the characteristic and differentiate the sample from others, okay? And as previously described, its time is often, it's difficult to define a species by morphological characters only. So when this happens, some genetic information kicks in to help this identify what these species are. Genetic information is sometimes used as basis for distinguishing the species. As you see, you'll see after the sequencing, you see the variation between species. You can also line them up in the phylogenetic tree to figure out what are the neighbors of this species and how, which level of taxonomy we can go down based on genetic information. Also, in addition, when it comes to an organism with a weak swimming ability, the habitat in which they live can also be useful information for distinguishing organisms. In this case, biogeographic information is very useful to predicting evolutionary features. So once we identified, we have to store them. So individuals have been identified in this way are stored in the storage as specimen. As I mentioned before, they are stored permanently because these specimens are defining species. They are significantly unique and has a great value. So let me briefly summarize what I have said in this presentation. So it can be said that taxonomy simply identifies living organisms, but all the technique in biological research can be used for classification. So as I said earlier, it takes a lot of experience and time to become a skilled taxonomist. But don't be afraid too much. Every maniple skill. You need some learning curve and time and patience. Okay, finally, taxonomy can most easily compare to a librarian. There are several criteria for classifying books. They contain a lot of knowledge, as you see in the left side of figure. Likewise, there are many ways to classify a living organism, from simple criteria to criteria based on the biological information or more. In addition, these specimens are information about the species and as used as a reference to follow-up studies. Taxonomy is an important field of study that can tell about the existence and changes of living things, living organisms, and to mankind now and in the future, based on very strong evidence, which is specimen. That's why we collect and preserve that specimen at MAVIC. Thank you for listening. All right. So it's 12:23. We'll just have. If you have any question, this is time. You can ask some question to Dr. Beck about taxonomy. In this process, if you have a question, I'll translate it to Dr. Beck to Korean, and he will deliver an answer, and I'll return the answer to you in English. So the process might be a little bit slow down the discussion, but thank you for understanding in advance. So if you have any question, raise your hand. On the Zoom app, there's a response button, and there is a raise hand button. If you have a question. I'm sorry. So if you don't have any question, why don't you watch a video? This video is about what MAVIC scientists perform as a biodiversity survey. It's about 20 minutes long or so. The presentation made by Luciana is more about the deep sea activity. This one is more like offshore coastal area. Even though the coastal area is different between Fiji and Korea, you will get the idea what kind of activities is performed for biodiversity activity, from collecting and identification and the storage. And after this video, if you guys have no further question, we'll conclude today's program.
right, so did you guys enjoy the video? All right, so that's how our Mavic scientist performs on the field. Okay, so hopefully you guys come over here and learn with them, and someday next year, hopefully. Okay. Without any further question, then probably we can conclude today's program, and we will meet 1 p.m. tomorrow. Oh, sorry. 1 p.m. right? 1 p.m. tomorrow. We'll come back with gender mapping and biological information tomorrow. So with that, I'd like to say bye and see you tomorrow. Bye.